In a groundbreaking discovery, researchers at the University of California, San Diego, have used artificial intelligence to uncover a previously unknown cause of Alzheimer's disease and potentially identified a new treatment approach. Published in the prestigious journal called Cell in April 2025, this research gives new hope to the millions affected by this devastating condition. Alzheimer's disease affects 1 in 9 people over 65, making it the most common cause of dementia worldwide. While we've known about certain genetic mutations that can cause Alzheimer's, these account for only a small percentage of cases. The vast majority of patients have what scientists call spontaneous Alzheimer's, and until now, its causes have remained elusive. The journey to this discovery began when the research team, led by Professor Sheng Zhong from UC San Diego's bioengineering department, identified a gene called phosphoglycerate dehydrogenase, or PHGDH, as a potential blood biomarker for early detection of Alzheimer's. In follow-up studies, they observed something fascinating. The expression levels of the PHGDH gene directly correlated with changes in the brain associated with Alzheimer's progression. The higher the levels of protein and RNA produced by this gene, the more advanced the disease appeared to be. Intrigued by this consistent correlation, the researchers asked a crucial question: could PHGDH be more than just a biomarker? To find out, they conducted experiments using mice and human brain organoids, miniature lab-grown brain-like structures. The results were remarkable. By altering the amounts of PHGDH expression, they observed consequential effects on Alzheimer's disease progression. Lower levels corresponded to less disease advancement, while increasing the levels accelerated the disease. This established that PHGDH is indeed a causal factor in spontaneous Alzheimer's disease. But how exactly does this gene cause Alzheimer's? Initially, the researchers hit a wall. PHGDH was only known to create an enzyme important for producing serine, an essential amino acid and neurotransmitter. However, experiments designed to connect this function to Alzheimer's repeatedly failed. The breakthrough came when the team turned to artificial intelligence. Using AI to visualize the three-dimensional structure of the PHGDH protein, they discovered something unexpected: a previously unknown substructure that resembles a DNA-binding domain found in transcription factors, proteins that regulate gene expression. This discovery was only possible through modern AI's ability to precisely map the protein's structure. The similarity was purely structural and not evident in the protein sequence, making it virtually impossible to detect through traditional methods. Further testing confirmed that PHGDH has a moonlighting role, a secondary function completely independent of its known enzymatic activity. Through this newly discovered mechanism, the protein can bind to DNA and activate specific target genes, throwing off the delicate balance of gene regulation in brain cells. This disruption triggers a cascade of problems that eventually leads to the early stages of Alzheimer's disease. In patients with Alzheimer's, increased production of PHGDH protein in the brain amplifies this disruptive effect. With this mechanism identified, the researchers sought a way to intervene. They found a small molecule called NCT503 that showed potential. While this compound doesn't significantly inhibit PHGDH's primary enzymatic function, which is important for normal cellular processes, AI modeling revealed that it could block the protein's newly discovered DNA binding capability. Crucially, NCT503 can also cross the blood-brain barrier, a critical requirement for any Alzheimer's treatment. When tested in mouse models of Alzheimer's disease, the results were promising. Treated mice showed substantial improvement in memory tests and reduced anxiety, two symptoms often associated with Alzheimer's in humans. Unlike many current treatments that target the buildup of beta-amyloid proteins, often referred to as plaques, this new approach addresses an upstream cause of the disease. By preventing the initial disruption in gene regulation, NCT503 could potentially reduce amyloid plaque formation in the first place rather than trying to remove plaques after they've formed. Another advantage is that small molecules like NCT503 could potentially be developed into oral medications, unlike current treatments that require infusions. The researchers acknowledge certain limitations in their study. There is no perfect animal model for spontaneous Alzheimer's disease, so they could only test NCT503 in available mouse models, which typically carry mutations in known disease-causing genes. The next steps will involve optimizing the compound and conducting FDA-required studies to move toward potential clinical trials in humans. This research represents a significant advancement in our understanding of Alzheimer's disease. By using artificial intelligence to uncover a previously unknown function of the PHGDH protein, scientists have identified both a cause of spontaneous Alzheimer's and a potential treatment approach. While much work remains before this discovery can be translated into clinical treatments, it offers new hope for developing more effective therapies for this devastating disease. As Professor Zhang noted, there may be entirely new classes of small molecules that can potentially be leveraged for development into future therapeutics. This research was published in the journal Cell on April 23, 2025, by researchers from the University of California, San Diego. The work was partially funded by the National Institutes of Health. For more information on this research, visit the links in the description below. If you found this video informative, please like, subscribe, and share with others who might be interested in the latest advances in Alzheimer's research. Thank you for watching.